Dr. Yasir earned his MPhil in microbiology as a gold medalist from the University of Health Sciences in Lahore, Pakistan. He then went on to earn his PhD in molecular microbiology from the University of Birmingham, where he studied the regulation of virulence genes in E. coli. Dr. Yasir is a busy researcher. It's really fantastic to see how much he's involved with. To name a few of those things, he contributed a really important role during the COVID-19 pandemic. He was a part of the COG UK team. And I actually met Dr. Yasir a few years ago, where his passion for supporting research in Pakistan and Pakistan-based researchers was a major driving force behind GetGenome's call for projects in the country in 2023. And we supported a cohort of Pakistan-based researchers sequencing over 100 important bacterial strains. So Dr. Yasir was really, really pivotal, central to that effort, and I thank him for those efforts. And today, Dr. Yasir is going to present a really intriguing piece of research. His talk is entitled Break It to Make It, TraDIS-Xpress, the story of random destruction to systemic understanding. So I'll hand over to Dr. Yasir and thank you once again.
Thanks a lot, James, for kind introduction. And thank you for your support throughout the call for the Pakistan 2023. That was a fantastic collaboration. I really appreciate that. So thank you for joining me today. Today, I'll talk about how we break the things to make something better. We are part of Norwich Research Park. So Norwich Research Park is a cluster of research institutes, university, and Norfolk University Hospital. So I'm based here at Quadram Institute, and James is at the Sainsbury Lab, GetGenome, and there's Arlem Institute, that is Bioinformatics Institute, and John Innes Center is the oldest plant institute in UK. And here is the University of East Anglia. And next to us is Norfolk and Norwich University Hospital. So here we have a fantastic cluster of scientists that are working on a range of projects from human health to plant and environmental sciences. So in today's conversation, I'll talk about the background of AMR and then the tools we are developing to understand the mechanisms of AMR and other biological mechanisms. And I'll give a few examples of the application of the tools that we have developed and we are developing, and a few variants of the TraDIS-Xpress, that's one of the tools. So I remember when I was a young biomedical scientist, we used to detect AMR doing PCR. So about 2 or 3 decades ago, when PCR came into practice, it was the most popular technique to find antibiotic resistance genes in the bacteria and do a PCR and find out if the gene is present, then that bacteria was labeled as resistant to that particular antibiotic. And then if there's a phenotype as well, if the gene is present, and mostly it's on mobile genetic elements like plasmids or phage, and if it's showing the phenotype, then we relate the genotype with the phenotype. But there were some cases when the phenotype was present, but AMR gene wasn't present. So that was the most interesting part for me, because I wanted to understand how the bacterial core genome has contributed to the resistance of this particular phenotype, or bringing up this phenotype. So the methods used by scientists from decades is, okay, let's mutate a gene and find out a phenotype. And if there's a phenotype, then we can relate that this particular phenotype or function is related to this gene. And there are 2 ways of mutating the gene. One is targeted mutation and one is random mutation. So The targeted mutation includes lambda-red recombination, like gene doctoring method or CRISPR-Cas method, where we target particular mutation or particular knockout of a gene. On the other hand, we have random mutations, like if we have a chemical treatment of the genome, then we can introduce mutations, but those mutations are random, they're not controlled ones. And same as the transposon mutations, like transposons are jumping genes that can insert anywhere in the genome. And Barbara discovered that mechanism in 1940s when she found the kernel color is different in maize, and it's because of the mobile genetic elements. And since then, they have been discovered and found almost all forms of life, from bacteria to humans and the plants and fungi and everything else we know of. So we use this transposon mutagenesis random mutation technique to mutate the genome and find the functions of the gene. So just explaining, before explaining to the method, I'll just explain two basic concepts. One is like essential gene and what is non-essential gene. So when a gene is mutated and the bacteria cannot grow, that gene is essential for the growth of bacteria, and we call it essential gene. And on the other hand, if we mutate a gene and the bacteria still can grow, then we call that gene as a non-essential gene. So we can make transposon mutant library by doing one electroporation. We can make thousands of mutants, and we can select them on plate. And then we can make many plates and collect a mutant library that can be of 1,000,000 mutants or 500,000 or 100,000, whatever our research question is asking for us to do. And here the interesting part starts. Then we can find the essential genes and the non-essential genes of the bacteria, and we can harness this technique or this power of the mutagenesis in our favor. So here's an example, like how we find out where the insertion has happened, because in a community where we have, let's say, 100,000 or 1,000,000 mutants in one tube, we need to find out where those transposon has been inserted. So for that, we do not do whole genome sequencing. We do just targeted sequencing, starting from the transposon to the outer genome. And this sequencing could be 50 base pair in length or 75 base pair or 100 base pair in length. And that would answer the question. So what we do is we extract the DNA and we design a customized library in such a way that sequences in such a way that starts from the transposon and comes out into the neighboring genome. And we used Illumina at the moment, but you can use any other sequencing platform as well. And here you see, like the yellow part is representing the part of the sequence that's coming from the transposon, and the red part is representing that's coming from the genome. And bioinformatically, we can remove the yellow part and then find on the whole genome where it aligns. And that would indicate the insertion site. So we can do it for all the mutants. And you can see that when some of the red arrows are indicating the insertions are on top strand and others are on the bottom strand, that is indicating the insertions of the transposons happening on the top or the bottom strand. And we can represent this without arrowheads as well, like lines. So in this case, the height of the line is indicating the number of the mutants in the population, and the location of the line is indicating where the transposon has been inserted, and red and blue are indicating if the transposon is on the forward or on the reverse strand. And we can show them on the same side of the genome as well, just for our understanding. Now we know that blue means the transposon was inserted on the bottom strand, and red means it was inserted on the top strand. So the real data looks like this. Here we can see that there are some gaps here, and these are representing essential genes. So this is the plot file that was created after aligning E. coli genome to the E. coli reference genome. And here, gray boxes or arrows are indicating the genes, and the blue are representing the proteins. And these insertion sites are representing if there are transposons, mutants are present in the mutant library that we used in this particular experiment. And here we see that DNAX and HEMH gene is essential for the bacteria to grow in this particular condition. And there are many genes that are non-essential, and the bacteria can tolerate mutations in those and still survive. So this is great, but like, what comes out of this? We can use this method of essentiality finding to understand the mechanism of action or resistance under different stresses, for example, antibiotic stresses. So what we do, like we made the transposon mutant library, and then we can store it in -80 freezer, for example, and we can take out the aliquot of the library of interest, and then we can grow it without stress as a control, and we can grow it with stress. In this case, for example, antibiotic, and we can find find that if there are genes essential or non-essential in this stress condition. So here we can see that gene B becomes essential in this particular stress condition. It was non-essential in the control, but it becomes essential. But a challenge lies. So gene A, it was essential in the control as well. And as it was essential in the control, we do not know if it is contributing to the stress or not. And if it were contributing, we would not be aware of that because there are no insertions at the first place. So there will be no decrease or increase insertions would have happened in this gene. So get around this challenge of not assaying essential gene. We have introduced this promoter, outside promoter. So what it does, it overexpresses the essential genes if they are playing any role in the stress condition. So why it was important to assay essential genes? Essential genes comprise about 7% of E. coli genome, and most of the essential genes are targets for the antibiotics or the most anti-infectives. So if they are playing any role, we are not aware of that, and that is very important for the understanding. So in the TraDIS, like a transposon-directed insertion site sequencing, there was only one outcome that was it was mutating the gene. But in TraDIS-Xpress, where we have promoter, it does inactivate the gene, but it also overexpresses the gene if that's important and we cannot mutate it. And also it represses the gene if it is detrimental and we cannot mutate it. And that's how we would know that this essential gene is detrimental for the stress condition. And it does through messenger RNA neutralization. So in short, TraDIS has only two outcomes, that is protection and knockout. And here the top plot is showing no stress or the control, and the bottom graph or the plot is showing triclosan stress. And here we can see that these two genes have less number of insertions as compared to the control. That means these two genes are being protected. And here, one gene that has more insertions as compared to control, that means this gene was detrimental for the bacteria and it's being knocked out. And here's an interesting example where this gene was essential, but it's being overexpressed here because it is one of the targets of the drug. And if there are more copies of that, then there are more chances of the bacteria to survive. And here's an interesting example of repression when RPON gene is being repressed because that's detrimental for the bacteria to survive in this particular stress condition and the phase of the growth. So I have explained to you TraDIS-Xpress, and I'll go through few applications where we can use it and how can we use it. So TraDIS-Xpress is a tool that is a functional genomic tool. Basically, it tries to understand the function of the gene. And we can use it to almost any process understanding or the mechanism understanding. So here are a few examples. Microbiome. Microbiome is a complex community where we can use TraDIS-Xpress to understand what is going on in the community and how the bacteria are interacting with the community members and other microscopic species and genera. And probiotics, we can use TraDIS-Xpress to understand the genes that are important for the delivery of bacteriocin or any other compounds of desired nature that we want. And anti-infectives that I'll talk about today, the TraDIS-Xpress can use to find novel targets and synergies, and it can be used to find the mechanisms of resistance from the existing antibiotics. And bioprocessing, it can be used to engineer strains that can deliver the product of interest in better quality and in better quantity. So here are a few examples of our publications that we did in the last few years. And I'll give a couple of examples here. So the experimental design for most of the publications was we used 4 different concentrations of the antibiotic, and we used untreated control with no stress. And as we have outward-facing promoter, so we used IPTG for the induction. And there's a molecule that turns on and off turns on the promoter, and we use 2 different concentrations of IPTG. And we extracted the DNA. So most of the papers or the research we have used this for concentrations, but in some examples we used more concentrations wherever we found that it's more interesting and necessary. For example, in I always kind of try to give the example of this marathon, because these mutants are like the marathon participants. So we let them run for overnight. And after that, we find out, okay, who are the winners and who are the losers, who came first and who could not make it. And that's how we would know that, okay, the mutations in those genes enable or disable particular mutants, and that's how those particular pathways are important for the survival. So in this example of triclosan, we used 8 different concentrations of triclosan stress, and we compared it with the control. And at 0 hours, almost all of them had grown, but at 24 hours, you can see that there are, at higher concentrations, there's hardly any growth. So on the right-hand side, we find, after the analysis, it's a heat map of the log-fold change of the mutants. And you can see that there is a different cluster of the genes at higher concentrations of stress as compared to lower concentration of stress. And that can be seen in the network analysis more clearly. So you can see at the lower concentration, the genes that are required to survive, they are different. So bacteria employs different strategy to grow if there is less stress or if there's higher stress. So depending upon the attack, they're very, very smart. So they use the resources very carefully. And that's why at the higher concentrations, all the genes are clustered together, and the lower concentration, the genes are clustered together. So here's an example of one of the essential genes that I want to give. Like we did include outward-facing promoter, and is it really working or not? That was one of the first questions. And Fabi is a non-target for triclosan. And when we did that triclosan stress as compared to control, you can see there are more insertions in blue color. That means the transposon inserted at the bottom strand, as we discussed earlier in that talk. And that means these all insertions are overexpressing this Fab basically. And there is no red insertion, so it's not knockout. So only the blue insertions are present. That means all the insertions are in one direction, and they are all empowering the over expression of Fab. And that was a very good example, and it gives us the confidence. And then later on, we find out there are essential genes involved in many other antibiotics, including meropenem, colistin, and gentamicin. But yeah, like this was one of the examples I wanted to give here. And another example I want to give is efflux pump. So efflux pump is a pump like that works in such a way it pumps out any unwanted molecule out of the bacteria. So here is the inner membrane, is the outer membrane, and the periplasm. And this is ACRB and ACRA. They combine with Tol-C and they pump out any drug or unwanted molecules out of the cell. And in this example, the unwanted molecule was triclosan, and they were pumping it out, and that was indicated in our data. So this is the genomic layout of that. ACRB and ACRA operon is located downstream of ACRR. That is the regulator of these two genes. So if it is present, then these two genes, they are repressed, they are not expressing. But if it's knocked out, then these genes are overexpressing. So what is happening here is a beautiful mechanism. So this ACRR is being knocked out. At the same time, it's overexpressing this ACRA and ACRB. That means there are more of these proteins are being made, and that's required for the bacteria to survive under the stress. So we did verify by that growth curve, and in the wild type BW25113 and the mutant ACRB, you can see that with one mutation only of ACRB, the bacteria could not export this triclosan, and it dies, while the wild type, it has the ACRB gene intact, and it survives under that stress concentration. So we did find all the previous stuff, of course, but we were interested in the novel mechanisms. So it wasn't known how triclosan is imported or transported inside the cell. And we find out that potassium channel is being used by triclosan for the transportation. And that was the first time that we find out that, okay, it's using this channel of potassium and it's being imported. And then, of course, then it acts on Fab that is involved in the phospholipid formation. And that's one of the examples of the TraDIS-Xpress power. So I want to talk about the synergistic combination. So amazing that it can knock out the genes and find the gene functions, but how can I use it for the other things? So an example is synergistic combinations. We did publish this paper in 2021, I think. It's a Microbiology Society journal. So what we did was we found in triclosan, in trimethoprim, so there was a TDK gene that was being protected. And on another screen, we were doing on zidovudine, we find out that TDK gene was being knocked out. So there was two opposite effects that we were seeing. And when we combine these two drugs, we find out they have the synergistic combination. They have the synergistic effect. And the trimethoprim MIC came down many times. So this is one example. And then we did find the similar kind of things in other studies as well, like in meropenem and cefzoludin. We find a similar kind of combination, like if one gene is being protected in one agent stress and it's knocked out in the other agent stress, there is most likely chances are they are having the synergistic combination. So I believe I would have shown you 3 examples, like how we can disrupt genes to understand gene functions. And we can harness the power of random mutagenesis for systematic understanding. So next, I think I'll give you a snippet of how we are using this TraDIS-Xpress for understanding the communities, how the communities are interacting. So the principle we are using is the same. So we have a gut model and a micrometrics. Micrometrics is a small instrument that mimics the environment of gut, all the gases and the environment that's required, and of the media, of course, we provide, and it mimics the gut model situation. And then we grow the library of our interest or the bacteria of our interest in the gut microbiome, and we try to understand how they're interacting with the gut microbiome and how gut microbiome is responding to the novel species or strain coming in. And we are using the same principle. We introduce a mutant library, and then we count the number of mutants, if they are reducing or if they're increasing. And that's how we deduce if certain number of mutants are increasing or decreasing. Then we kind of deduce and find the pathways that are required to survive in that condition. And similarly, we do the population genomics at the same time, and we see the changes happening in the population of the microbiome and how the microbiome is reacting to that. So we can use transposon mutagenesis to identify synergistic combinations by studying gene functions under stress, and we also find we can find the microbiome interactions by studying the mutant library in the complex environment. So as I have discussed with you guys, we are using the mutant library, not a single mutant. We are using all the mutants in one container. That poses certain kind of challenges. So if there is a tug of war between two teams, and one team wins, but that does not necessarily mean each and every member of the team is winning from each and every member of the other team. So some of the team member would have unfair advantage over the other ones, and that there will be uneven match. And that is exactly true for bacteria. So when we are growing a community of mutants, there are certain mutants that might not be able to produce certain intermediary molecules, but they might be borrowing it from the community that's been excreted into the media or the environment, and they might survive. But when they're growing in a single vial, they might not be able to grow. So to understand that, the ideal situation would be when we can grow a single mutant in a single tube. And just imagine growing 500,000 mutants and 500,000 tubes and the manpower needed. That's massive. So the solution to this kind of problem is droplet genomics or microfluidics. So here's one of the examples of the instrument that's used. There are two syringes. One brings the oil and the other brings aqueous solution. And here is the chip that is shown. So these two lines, they are representing the groups that are bringing oil, and this one is bringing aqueous solution that contains bacteria. So it brings oil, and this brings aqueous solution with bacteria, and it combines them in such a way that it forms a droplet. So here you might be able to see, if I can play this video. Yeah. So these oil is coming from this side, and the aqueous solution is coming from here, and it is converting them into droplets, and each droplet should contain one bacteria. And we create these microscopic droplets. Excuse me, sorry. And we can store hundreds of thousands of these micro droplets into a microfluidic tube. And that's how we can study mutants into their individual environment while we are still using one tube. So we did that with the droplet genomics of the E. coli library, and we studied the stress of ciprofloxacin. And we find out there are 41 unique genes that are required to survive under ciprofloxacin stress when they are grown in the droplet genomics. as compared to the control that was without any stress. And we also, of course, did the community stress, or the stress that we are using already in standard lab format. And here are a few examples I have given. So we use 3 different concentrations, half MIC, 1 MIC, and 2 MIC. For the droplet genomics, we used only half MIC. And as you can see, there is a very less overlap between the 2 forms of stresses. And these are the 3 common genes that are present among all 4 stresses, 1 of the droplet genomics and 3 community stresses. MAR is a regulator, and it regulates many genes that are related to stress. So we were interested, like, okay, if there are not present in all the stresses, but anything common with the droplet genomics. And we find out that all the stress that we get, genes we get, they are going in the same direction, irrespective of it is droplet genomics or a community stress. Or the community environment, in the community environment stress. So you can see that either they are in the blue, that means they're being knocked out, or red, they are being protected. And also I want to point out all the genes that we are seeing in the droplet genomics, they're more looking like a 2 MIC instead of like half MIC. So it means the impact of MIC could be different when they are being in the different environment, like when they are the mutants are alone as compared to in the community. So I have just showed you an example of like how we can use droplet genomics to examine individual mutants in the community settings. So with that, I think I'll stop here, and I'll leave some time for the question and answers. And I do want to acknowledge a lot of people here. Professor Ian Charles and Professor Mark Webber I work with, and Keith Turner, who helped a lot in the lab and library formation. And Sarah is our bioinformatician, and my other colleagues and collaborators. And I do want to thank our funders, BPSRC and MRC. And Center for Microbial Interaction, that is the center we have developed at Norwich for all the microbiologists across all the institutes and the university. And of course, GetGenome for giving me the chance to present. And if I could not answer your question right here, you can email me, and I hope I'll reply as soon as I can. Thank you very much for listening.